Hello everyone, welcome to our tutorial on genetics. These questions, as you know, we have already seen them in class. So the purpose of this video is for us to revise the techniques that we use when we do a Punnett square and we try to solve genetic questions. So let's read the question together. A heterozygous round seeded plant. Okay, big R, small r. Obviously, whenever you hear the term heterozygous, you have what we call big R, which is the dominant allele, and small r is the recessive allele. So in the test, this will not be given to you. You will have to know what is heterozygous, homozygous, and so on. Is crossed with a homozygous round seeded plant. So homozygous, here it's big R, so obviously it's homozygous dominant because you have two dominant alleles which are big R, big R. Now the question is as follows: what percentage of offspring will be homozygous? Okay, so basically your answer here should be in percentage. Okay, so let's begin this question. For you to begin this question, the first step always is to write down the alleles. Okay, so how are we going to do this? So big R is the allele for round seeds. Small r is the allele for non-round seeds. Okay, now the next step, that's step 1. The next step is reading the question again. So when it's telling you that a heterozygous round seeded plant is crossed with a homozygous round seeded plant, basically here we are talking about the parents. So parental phenotype, as you know, phenotype is the physical characteristic. So the parental phenotype is going to be round seeds, right? Hetero cross with round seeds, homo dominant. Okay, after that you're going to write down parental genotype. This has already been given in the question, but sometimes it will not be given and you have to know about it by understanding the phenotype. So big R, small r cross with big R, big R. The next step is to write down the alleles. Okay, so basically we will have as alleles big R, small r cross with big R, big R. Remember that your alleles have to be always circled. Fourth step is doing our Punnett square. So Punnett square. Okay, so when you do your Punnett square, remember that one of the parents will go on top here, right, horizontal, and the other parents will go down here, vertical. Okay, so you're going to have big R, right, this and this one together. Again, big R, right, then big R, small r, then R, small r again. Now after that, step 5, you're going to write down the phenotype of the offspring. So you can write offspring, right? Phenotype. And what is the phenotype of the offspring? So big R, that's dominate, homo, round, right? What else do we have? We have hetero, dominate, right? Round. Okay, now let's continue. So you have the offspring phenotype, now the genotype, which is already given, or you have just found from the Punnett square, big R, and big R, small r. So if we go back to the question, what has been asked? Okay, so let's go back to the, so if you look at the question, you want to know what percentage of offspring will be homozygous, big R, big R. For you to know that, right? So let's write it down. Step 6, percentage of homodominant. Remember, in the test you have to write full sentences, right? So how many are dominant? 1, 2, right? And out of how many? 1, 2, 3, and 4, right? So 2 out of 4. This gives us half, and if you use your calculator, you will get 50%, which is the answer. So you box your answer. Here you have to be very careful. Remember that your alleles have to be always circled, and all these steps have to be present, or else you will lose marks on the test.